Hi everyone, I'm Chen Zixia from CSBL. Today the title of my presentation for the journal club is A Tripartite Microbial Co-Culture System for the Novo Biosynthesis of Diverse Plant Phenylpropanoid, published in Nature Communications in 2023. Phenylpropanoids are a very important family of secondary metabolites in plants. Their common structural features is an aromatic ring connected to a 3-carbon side chain. Their synthesis starts from tyrosine or phenylalanine through a deamination reaction. Cinnamic acid is generated, and further hydroxylation forms p-coumaric acid, which is an intermediate metabolite we focus on a lot today. We can see that p-coumaric acid is one of the core hubs of the entire phenylpropanoid metabolic network. Starting from p-coumaric acid through a series of oxidation, methylation, and condensation reactions, it can lead in multiple directions to form a series of natural products with different structures but significant functions, such as flavonoids, coumarins, lignans, and phenylpropines. Among the phenylpropanoid compound, eugenol possesses multiple biological activities, such as antioxidant, anti-cancer, and anti-obesity properties. However, its content in plants is usually very low, purifying is very difficult, and the process is restricted by the season and environment of the plants. Therefore, in this thesis, through synthetic biology methods, this pathway was reconstructed in microorganisms, starting from simple sugars. A stable, efficient, and controllable biofactory was constructed. Module co-culture brings flexibility and diversity to the synthesis of polyphenolic products. However, co-culture systems still face problems in practical applications: insufficient system stability, metabolic toxicity that can affect yields, and difficulty in reusability, and so on. Therefore, this paper introduces the ELM platform to address these issues. ELM is a printable, photocurable, and reusable active biomaterial formed by encapsulating microorganisms in F127BUM thermosensitive hydrogel. The 3 key steps of ELM are gel preparation, gel storage, and on-demand production. In the gel preparation stage, a solution containing microorganisms is printed or injection molded and cross-linked and certified with ultraviolet light. The certified active hydrogel is then transferred to a culture medium for the cell growth. In the gel storage stage, the grown active hydrogel is stored and proceeds in different ways, such as refrigeration, freezing, which is convenient for long-term storage, transportation, or batch-by-batch use after large-scale preparation. In the on-demand production stage, when needed, the stored gel is rehydrated and placed in a fresh culture medium. This material can be used repeatedly for multiple rounds. After being rinsed with PBS, it can enter the next round of production. Ultimately, the standardization, reusability, and industrial deployment of the microbial platform are achieved. This figure shows the overall metabolic design concept. The eugenol biosynthetic pathway is divided into 3 modules. Module 1 enhances the tyrosine metabolic flux and synthesizes p-coumaric acid. Module 2 converts it into ferulic acid. Module 3 further converts ferulic acid into eugenol. Each module contains key heterologous enzymes such as Flavobacterium johnsonia (TAA), Saccharomyces asparanesis C3H, Petunia hybrida EGS, and so on, which are distributed in different strains to reduce the metabolic pressure on a single strain and inhibit the formation of byproducts. Figure 2a shows the single-strain strategy constructed by integrating all metabolic modules into one E. coli strain. Figure 2b presents the co-culture scheme in which Module 1, Module 2, and Module 3 are placed in three different strains respectively. The HPLC chromatogram in Figure 2c indicates that the single-strain approach cannot effectively produce eugenol, while the three-module co-culture system detects a distinct eugenol peak at the target retention time, confirming the effectiveness of the co-culture strategy. After demonstrating the effectiveness of this three-part co-culture method for de novo eugenol production, the yield of ferulic acid was then optimized. The researchers first optimized Module 1 and Module 2 to maximize the production of ferulic acid and minimize the accumulation of coumaric acid using a median optimization strategy. Figures 3b and 3c evaluated the effect of different nutritional additives on the intermediate product and found out that supplementing with iron, pantosanic acid, niacin, riboflavin, and gluconate was helpful in increasing the yield of ferulic acid. The heat map in Figure 3d and the bar chart in Figure 3e indicates that at Module 1 to Module 2 ratio of 1 to 3, the yield of ferulic acid reaches 363 mg per liter. This is the highest reported ferulic acid titer to date. Figure 4a shows that Module 3 is split into two sub-modules. One converts ferulic acid to coniferol alcohol, and the other converts it to eugenol. The conversion efficiency of the two sub-modules was verified respectively. Likewise, the combined Module 3 strain was suitable for converting ferulic acid feed to eugenol with minimal buildup of the coniferol alcohol intermediate. Figure 4b demonstrates the impact of different Module 2 Module 3 ratio on the yield. Figure 4c and 4d were verified in a 1 ml deep well plate and a 30 ml shake flask respectively. The highest eugenol yield of 66 mg per liter was achieved at a ratio of 1:3:1, marking the first complete de novo eugenol synthesis starting from glucose. To demonstrate the versatility of the platform, the researchers rapidly achieved the synthesis of 3-phenylpropene products through changes in module combinations. In Figure 5a, the complete Module 1 to Module 3 were used to generate eugenol. In Figure 5b, COMT in Module 2 was removed to produce hydroxycavicol. In Figure 5.3, the entire Module 2 was removed to produce cavicol. This shows how to utilize the modular structure and the substrate selectively of the enzyme to regulate the types of products. Figure 6a shows the HPLC detection result. The green, blue, and purple lines represent the retention times of hydroxycavicol clavicle, and eugenol respectively. Figures 6b, 6.3, and 6d are the corresponding AOC mass detections, verifying the characteristic mass-to-charge ratio of eugenol, hydroxyclavicle, and clavicle. Through these chromatograms, it is further confirmed that different module combinations have indeed achieved the de novo synthesis of the desired target compounds. Figure 7 demonstrates the advantages of the ELM system in product synthesis, stability, and reusability. In figures 7a to b, for the synthesis of ferulic acid, the ELM systems with separate cultures and mixed culture are compared with the liquid co-culture. In figures 7c to d, for the synthesis of eugenol, the results shows that only the ELM system with a separate culture can achieve product synthesis and can be reused after being washed with PBS. These results indicate that the ELM platform significantly improves the system stability and reusability. This study has achieved for the first time the de novo synthesis of multiple plant phenylpropanoid products, including eugenol, demonstrating the potential of modular microbial co-culture combined with ELM encapsulation technology in natural product synthesis. In the future, this platform can be extended to more metabolic pathways such as flavonoid and lignans, and methods like machine learning can be integrated to further optimize the module design and combination strategies. That's all. Thanks for your listening.